We're very pleased today to have with us Gabriela Ramirez-Hernandez. She is presenting inhibition of formate production blocks CD8 positive T cell responses and delays disease onset in a mouse model of type 1 diabetes. She's coming to us from the Harvard T.H. Chan School of Public Health and the Broad Institute of MIT and Harvard. And a little bit about Gabriela. She earned a bachelor's degree in pharmacy and a master's degree in neurobiology. She completed her PhD in biomedical science at the National University of Mexico, where she studied the protective effects of the hormone prolactin against the chemical induction of type 1 diabetes, including the prevention of beta cell loss and pancreatic inflammation. She completed a one-year postdoctoral fellowship in Stephen Kisser's lab at the Joslin, focusing on cellular senescence as a driver of islet autoimmunity. And currently, she's a postdoctoral researcher in the Corey lab at the Molecular Metabolism Department of the Chan School of Public Health, where she's investigating mitochondrial metabolism in CD8 positive or T cell response during type 1 diabetes. I'm going to drop her recent preprint into the chat and we will take questions at the end. Thank you very much, Gabriela, for joining us. We're really interested in this paper, and it's very different than some of the ones we've seen recently. So we're excited to have you present it.
Thank you, Monica. Hello, everyone, and thank you for the invitation and the kind introduction. I also appreciate all the audience joining us today. I believe this is a great forum for sharing and networking about the latest insight in the type 1 diabetes field. Therefore, I'm excited to present our preprint work. Fingers crossed, it might be published soon. But our project focused on the inhibition of formate production, which blocks CD8 T cell responses and delays disease onset in a mouse model of type 1 diabetes. And essentially, the primary goal of this project is to identify new control points for CD8 T cell activation and expansion in the autoimmunity context of type 1 diabetes. I hope you enjoy it. Okay, located within the pancreas, the islet of Langerhans are the structural and functional units that produce crucial hormones to maintain glucose homeostasis. Pancreatic beta cells are one of the most abundant type of cells in the islet, and they are the only cells in mammals capable of producing and releasing insulin, which is essential for enabling glucose uptake in various tissues such as adipose tissue and muscle. During type 1 diabetes, beta cells are destroyed, resulting in the absence of insulin production that leads hyperglycemia. And the attack and destruction of pancreatic beta cells is mainly mediated by the CD8 T cells through a coordinated process involving their interaction with antigen-presenting cells, such as dendritic cells and macrophages, and differentiated CD4 T cells. The current immunotherapies for type 1 diabetes still faces challenges as its long-term effectiveness remains limited. And in order to improve the immunotherapies against type 1 diabetes, our approach proposed to target the activation and expansion of CD8 T cells to prevent or minimize the beta cell destruction and thereby reducing the progression of type 1 diabetes. But first, I would like to introduce you what is happening during the activation of T cells. So the T cell response involves metabolic reprogramming as cell shift from a quiescent catabolic state to an active anabolic state, supporting their ability to maintain a proliferative and biosynthetic phenotype. One of the hallmarks of this transition is a reprogramming in mitochondrial metabolism. And this metabolic switch includes aerobic glycolysis, glutaminolysis, and mitochondrial biogenesis, facilitating oxidative phosphorylation and the TCA cycle. The mitochondrial proteome analysis during this reprogramming of mitochondrial metabolism showed that one-carbon metabolism was the only pathway significantly induced during the early activation of CD4 T cells meaning 24 hours post their stimulation. And similarly, when we analyze the proteome exchange database, we identify one carbon metabolism as the most upregulated pathway among various mitochondrial processes in activated CD8 T cells. So now let me introduce you this metabolic pathway that is also known as the folate cycle. One carbon metabolism is a crucial pathway for proliferating cells, such as activated T cells, because this pathway supplies cells with one carbon units that are needed for the DNA synthesis and repair, the amino acid balance, and the redox defense. This pathway implicates the serine metabolism to formate within the mitochondria in the presence of tetrahydrofolate as a cofactor and incorporate the single carbon of formate directly in the synthesis, impurine, and methylation reaction. Serine deprivation, serine or glycine deprivation, as well as folate supplementation, has been one of the experimental strategies to control not only T cell expansion, but also cancer cells. And interestingly, an in vivo one carbon metabolism CRISPR screening in a lung inflammation condition identified the gene of the mitochondrial enzyme MTHFR2 as one of the key contributors to T cell proliferation and inflammatory function. Furthermore, MTHFR2 serves as a control point for the CD4 T cell response, regulating their proliferation, activation, and differentiation. And moreover, this enzyme was upregulated in several inflammatory and autoimmune diseases in humans, including arthritis, lupus, and sclerosis multiple. And the pharmacological inhibition of MTHFR2 decreased the severity of experimental autoimmune encephalomyelitis, inflammatory bowel disease, and arthritis, pointing out this pathway and specifically MTHFR2 as a key target for regulating autoimmunity or T cell response. However, the effect of the inhibition of MTHFR2 in CD8 T cells, which are the direct killers of pancreatic beta cells, and its implication in type 1 diabetes remains unknown. Therefore, our project investigates the role of MTHFR2 in the CD8 T cell response using in vivo and in vitro approaches, and with a specific emphasis in type 1 diabetes. So first, to confirm the positive regulation of one carbon metabolism during the activation of CD8 T cells, we measure the one carbon metabolism enzyme levels using a murine primary CD8 T cell. We observe upregulation in all of the enzymes with a special emphasis in the ones within the mitochondrial branch. And since these three enzymes contribute to the production of formate, which is a critical metabolite intermediate in this pathway for the DNA synthesis and repair, we determine the dynamic of formate production and secretion during the activation of CD8 T cells. We observe that the production and secretion of formate peak 48 hours and 24 hours post the activation or the stimulation of CD8 T cells. And using the strategy of pharmacological inhibition of MTHFR2, we were able to block these increasing in formate levels observed during the activation of CD8 T cells. Also, the inhibition of MTHFR2 regulates the function of CD8 T cells, as was illustrated by less cell viability or less cell survival, less cell proliferation, less activation of CD8 T cells, and more exhaustion of the CD8 T cells, meaning more dysfunction of these cells. In addition, the treatment with the MTHFR2 inhibitor decrease the capacity of the CD8 T cells to produce effector molecules such as the inflammatory cytokine interferon gamma. Then, trying to understand the mechanism implicated in this MTHFR2 inhibition condition, we know that the inhibition of MTHFR2 blocks the production of two crucial metabolite intermediates, 10-formyl tetrahydrofolate and formate. These two metabolite intermediates are needed or are essential for the formylation reactions during the purine synthesis, allowing that purine intermediates can produce the purines. So in other reports have shown that during the inhibition of MTHFR2, an accumulation of purine intermediates was observed. Therefore, we measure the purine intermediates levels in this condition. And what we found, it was a big accumulation in the intermediate GAR, but not in the rest of the other purine intermediates. This was important because other reports have suggested that the accumulation of ICAR trigger the AMPK signal that is a negative regulator of mTOR pathway, which is a critical pathway for the CD8 T cell response, or in general for the T cell response, targeting downstream molecules essential for the survival, proliferation, differentiation, and production of effector molecules in T cells. In line with a no observed ICAR accumulation in our model, we didn't see any increase in the signal of AMPK. Therefore, we look for another regulators of mTOR pathway that can be modulating under the inhibition of MTHFR2. And what we found it was that the REV and the KRAS mTOR regulator are downregulated in the inhibition condition, and in consequence, some of the downstream mTOR targets, such as S6, HIF1 alpha, and STAT3. So far, I have shown you that the inhibition of MTHFR2 modulate the CD8 T cell function by modulating or regulating mTOR regulator KRAS and REV, and key downstream targets for the essential for the T cell response, HIF1 alpha, S6, and STAT3. Then, since I told you that MTHFR2 decreased the formate levels, this is essential for this pathway, we wonder whether the supplementation with formate will be able to restore or reverse all this effect of the MTHFR2 inhibition. So we treat the cells or we supplement the cells with exogenous formate, and what we observe is the supplementation with formate restore or reverse the lower levels of formate observed in the presence of the MTHFR2 inhibition. And the cells that were supplemented with formate achieve the proliferative profile, such as in the control group. Moreover, formate supplementation restore the activation of CD8 T cells and also decrease the dysfunction of these CD8 T cells, as was illustrated by the expression of PD-1. And finally, the supplementation with formate restore the expression of REV and KRAS and downstream mTOR target HIF1 alpha, S6, and slightly phospho-STAT3. Then, in 2021, it was identified the most pathogenic population of CD8 T cells that can drive type 1 diabetes. This pathogenic population of CD8 T cells express or are specifically recognize the islet protein IGRP, and they also express higher levels of the transcriptor factor TCF1. This transcriptor factor is crucial for the CD8 T cells self-renewal, longevity, and memory. Also, these IGRP-specific CD8 T cells with higher levels of TCF1 have a progenitor signature, so therefore they were called like such as stem-like CD8 T cells. And the effect of this pathogenic population was so robust since the the transfer of a tiny amount of TCF1 high expression CD8 T cells was able to provocate hyperglycemia in lymphopenic NOD skid mice, in contrapart to the transferring of maybe 6 or 7 times more TCF1 low expressed with lower expression of TCS1 cells that were not able to provocate diabetes in these animals. So they suggest or they identify this population as one of the most pathogenic ones for driving type 1 diabetes. So having in mind these results, we wonder whether our inhibition of MTHFR2 could regulate the expression of TCF1s. And the short answer was yes. In the presence of the MTHFR2 inhibition, more of the cell express or the population of a TCF1 low expression shift a little bit to the in the presence of the MTHFR2 inhibition, and formate supplementation restore this proportion of high and low expression CD8 cell expressed TCF1. So in order to recap a little bit all our results so far, I have shown you that in the activation of CD8 cell in vitro, one carbon metabolism is one of the pathway, more upregulated pathway, with a special upregulation of their enzymes and the formate production and secretion. And when we inhibit MTHFR2, we decrease the formate levels, we block or decrease the expression of mTOR regulators KRAS and REV, and as a consequence, we decrease downstream mTOR downstream targets S6, HIF1 alpha, and STAT3. And the phenotype under this inhibition condition is less CD8 cell survival, proliferation, activation, cytokine production, and cell renewal, and more dysfunction of these cells. And all these phenotypes can be largely restored or prevented by the supplementation with formate. All these in vitro results allow us to propose that the treatment of or the inhibition of MTHFR2 could be a potential treatment for preventing or delaying type 1 diabetes in vivo. So for testing this hypothesis, we use the NOD mouse model. That is one of the well-established type 1 diabetes model in rodents. And the development of diabetes in these animals occurs at different stages. In the early period or the early stage in the life of these animals, we can observe the immune recruitment within the pancreas, the immune cell recruitment within the pancreas with no pancreatic beta cell attack. Then between the week 8 to 10, we are in the peri-insulitis stage, where the T cells invade the pancreas, but they are not attacking pancreatic beta cells. The active attack and destruction of pancreatic beta cells occurs within the insulitis stage. And then this triggers the diabetes or hyperglycemia state. This immunofluorescence image, I think, pretty illustrate the evolution of diabetes in these animals. In the early stage of the animals, we can see the islet with no evidence of immune cells invading the islet. And during the insulitis period, it's quite evident that T cells are invading and attacking pancreatic beta cells, decreasing the pancreatic beta cell mass. Okay, so our first question, it was whether formate could represent a potential biomarker that predict type 1 diabetes in this model. The short answer, it was no. At least when we measure serum formate levels, we didn't see any peak or increase in formate levels. In counterpart, we saw a decrease in formate levels along the development of type 1 diabetes. But we know serum formate levels can come from different tissues, and it's hard to explain the dynamic of serum levels. So then we measure formate within critical lymphoid organs essential for T cell development, maturation, and activation. That is the thymus, the spleen, and the pancreatic lymph node. And what we observe is a higher levels of formate in the pancreatic lymph node, where the activation of CD8 T cells occurs in the week 10. That is the week that precedes the insulitis stage. Therefore, our strategy was to treat animals, euglycemic NOD animals, starting with the MTHFR2 inhibitor, starting at week 10, where we observe an increasing in formate levels in the pancreatic lymph node, and where the insulitis stage just starts. And we treat the animals for 10 days. That is the safe period reported for this drug, 2 times per day by oral gavage. We found that the treatment by itself didn't affect the body weight of the animals, and the animals treated with the MTHFR2 inhibitor tended to have less glucose level. And when we measure the diabetes onset, we observe a decrease, a delay in diabetes onset and a decrease in diabetes prevalence or incidence in the under the treatment condition with the MTHFR2 inhibitor. And to try to understand this effect, we evaluate the insulitic lesions in the pancreas in these animals once they develop diabetes. What we observe is the insulitis score is less in those animals treated with the MTHFR2 inhibitor. And currently, we are trying to characterize the cellular phenotype of these insulitic lesions. In theory, most of the cells present in the insulitic lesions should be CD8 T cells and B cells, but we would like to just characterize this cells phenotype to understand in which cells this treatment have a higher effect. Okay, so the message that I would like to you take to home is that the inhibition of MTHFR2 blocks the formate peak that we observe is induced during the activation of CD8 T cell in mutant CD8 T cells, primary CD8 T cells. Then the inhibition of MTHFR2 modulates the CD8 T cell response, meaning their survival, proliferation, activation, dysfunction, and cytokine production, facilitate the GAR accumulation, and suppress mTOR downstream targets, HIF1 alpha, S6, and STAT3 through the modulation of mTOR regulators, KRAS and RAS. Also, MTHFR2 can regulate CD8 T cells TCF1 levels that could represent a novel role of MTHFR2 regulating the cell renewal and memory of CD8 T cells. And in vivo, the inhibition of MTHFR2 delays type 1 diabetes onset in this NOD mouse model. So then, our future avenues for this project. In order to evaluate the impact of this treatment in human, we are evaluating this treatment in primary human CD8 T cells to verify if we can observe exactly the same modulation if in CD8 T cell response in human conditions, right? So the second further investigation is in order to understand the direct effect of lacking MTHFD2 in CD8 T cells in vivo. We are planning the next experiment using the NOD animals. We want to collect autoreactive CD8 T cells, and we want to uh, delete the MTHFD2 enzyme in order to transfer these genetic modulated CD8 T cells in the NOD skid animals. These NOD skid animals have exactly the same genetic background such as the NOD animals, but they are lymphopenic. That means that they don't have any T or B cells, and they are diabetes free. If we inject or transfer a vehicle in these NOD animals, these animals, they should not develop diabetes or should not develop any autoimmunity, right? If we transfer in these animals autoreactive CD8 T cells, these animals develop diabetes or the autoimmunity due to the attack of the pancreatic beta cells for these CD8 T cells. And our hypothesis is that if we block or if we delete MTHFD2 in the autoreactive CD8 T cells and transfer in the NOD skid animals, we potentially be able to prevent the autoimmunity in this animal. And finally, the last avenue for this project is trying to identify the mechanism of mitochondrial formate export. When I introduced you this one carbon metabolism pathway, I show you that this pathway implicates the serine metabolism to formate within the mitochondria. Then formate need to be exported to the cytosol in order to be incorporated in the purine synthesis and methylation reactions. But the mechanism how formate export the mitochondria, the mechanism how formate that is produced in the mitochondria but need to be used in the cytosol, how this process happens is still unknown. So we hypothesize that if we can identify this mechanism, we also can identify another potential regulator of the CD8 cell response. And in order to address this question or this gap, we did a CRISPR screening strategy using control URCAT T cells and URCAT T cells treated with the MTHFD2 inhibitor. We use a metabolism library that is targeting 3,000 genes, and after 14 doubling, population doubling, we proceed with the deep sequencing of these two populations. And so far, we have some candidates for this mitochondrial formate transporter, and we are characterizing the phenotype of this candidate in the genetic deletion of these transporters that we identify. And in the future, we will exciting to check their ability to regulate CD8 T cell response. And finally, I just would like to thank all the team. This is not an individual project. This is a collaborative project with all the co-authors in the manuscript, but also all the support from Cori Lab and the Van der Heyden Lab in the MIT. And also, I would like to thank all the funding sources that have support this project. So, and now I'm happy to take any question.
Thank you so much, Gabriela. This is really some interesting work. And of course, we love to see this type of cross-lab collaboration. And I wanted to open it up to questions. If anyone has a question, please drop it into the chat or feel free to unmute yourself or raise your hand. got a lot of accolades clapping in your audience. Okay, while people are formulating their questions, I have one. Um,
Yeah.
can I guess what we can let's talk about translation. So several cancer groups, right? They're testing this MTHFD2 inhibitors clinically, right? Could
Mhm.
these or related agents be repurposed to prevent or delay type 1, or what safety concerns might need to be addressed?
Yeah,
Big
you
question.
mean yeah. You mean using this MTHFD2 inhibition in the clinic? You mean translating to the
Right, right.
clinic?
What's the path look like?
Yeah, uh, I think it's it's challenging because of course we know that the prevalence of the population where type 1 diabetes is more prevalence is in infants
Mm.
or
Well,
in young population. So
pediatric
using
population, yes, but there is an
exactly,
increase now in adults.
yeah,
So
yeah, as well.
possible.
But especially in infant or in young population, since they are still growing and developing, targeting MTHFD2 that is also crucial for those cells that are proliferating. Maybe so far it's going to be still challenging and maybe not like one of the best strategies for controlling um, type 1 diabetes. Could be like uh, a strategy that can control the CD8 T cell response maybe in adults or a concomitant treatment that potentiate another immunotherapies, but not as a single therapy, I will
No,
say.
no. I mean, I think right now the field is kind of talking about multiple therapies used in concert
Yeah,
or in, one after the next. But
yeah,
you can
mhm.
rescue it, right? You've talked about how you can rescue this with formate. So perhaps there's some kind of window opportunity there as people head towards type 1 diabetes that could be addressed. But I mean, obviously, you're looking at mechanism like why do these, T cells respond the way they do? And you are in the NOD mouse and the NOD skid mouse. So you're taking the first steps, but it was just kind of a big question. I guess you also have to consider that the one carbon metabolism is also active in pancreatic beta cells. So, could there be like risks for beta cell survival and stress resilience as you're progressing towards type 1 diabetes? Do you think there might be any biomarkers or metabolite profiles or even like a methylation signature or T cell subsets that could indicate that one carbon metabolism is actually driving autoimmunity in people at risk for type 1 diabetes? Or is there any kind of way of tracking this?
Yeah, at least I guess uh, should be. It's also hard because uh, where we observe this upregulation in hormone levels and also in MTHFD2 is in the pancreatic lymph node. So getting biopsy or the sample from that tissue is going to be a little bit hard or challenging for
Mhm.
translating this in humans. I strongly believe that MTHFD2 could be a potential biomarker or that predict
Mm.
the time when the CD8 T cells are more proliferating and in the expansion state. However, how to take the biopsy or how to have the tissue for analyzing or measuring the formate levels is maybe so far is still challenging.
Yeah, I mean, there are the samples, there's a very large and, well, relatively speaking, repository of samples, cadaveric samples, with NPOD and places like that that maybe are worth thinking about. But yeah, there's a lot of really interesting mechanism you've discovered here, and it will be really cool to see, sort of from the genetic side or the phenotype side, like what is that transporter in the mitochondrial, membrane there?
It is.
Right? And I mean, are people who are there some kind of genetic phenotypes, in people who progress to type 1 diabetes, and some endotypes of those that progress to type 1 diabetes that possess that kind of alteration? That'll be very cool. Okay, here's another question from City of Hope. Brian Liu, great work and talk. Does MTHFD2 impact iTreg and or nTreg?
In T-Rex, in CD4 T-Rex cells, MTHFD2 modulate also their expansion and their function. And interestingly, in CD8, in CD4 cells, the treatment with MTHFD2 turns to trigger the differentiation or increase the differentiation of CD4 T cells to Tregs rather than to Th17 cells. So apparently, this MTHFD2 can modulate the differentiation of CD4 T cells in favor to Treg cells. Also in Treg cells, MTHFD2 increase the oxygen consumption rates and ECAR or the glycolysis. So yeah, that is the effect in Treg. I don't know. I think no one have investigated the effect of regulating MTHFR2 in Tregs as a strategy for controlling pancreatic beta cell attack. I think that is an interesting new avenue for investigating. But at least, yeah, in the other type of cells, it said Tregs, and the other, I forgot the other type of cells. In the question, I don't know. In the question, I didn't, I forgot the other population. You said Treg
The
and
nTregs and iTregs.
got it.
He's got it in the chat if you want to take a look at it.
I cannot. I don't know why I don't. I cannot.
That's okay. Brian, you want to
Got it.
and ask? That's fine too.
Yeah. Oh, yeah. Okay. Yeah. In the iTregs, I don't have any idea, honestly. I'm not sure if someone have tested.
Okay. Well, we wish you the best in getting this preprint into a full-on paper, and I'm sure it's going to trend that way. It's a really interesting paper, and again, we're looking to see what your follow-up experiments show. So thanks so much for sharing this, and great effort. We wish you guys the best of luck, and yeah, keep going in these mechanistic studies. They're very important. Thanks so much.
Thank you so much, Monica. Yeah.
Thank you. Have a great
My
rest of your
pleasure.
day, Gabriela. Bye-bye.
Thank you. Bye-bye.